As part of the Crohn's and Colitis Young Adults Network Fellowship, I wanted to introduce you to my research. My name is Karen, and I am part of the 2022 Fellows. I am currently a second-year PhD student at the University of Florida studying biomedical engineering. This means that I actually conduct research, and my research project focuses on the effects of the human microbiome on inflammatory bowel disease and colon cancer. Some of the things that we do in our lab are culture bacteria as well as human cells. We occasionally also do DNA sequencing to confirm the identity of our bacteria. Doing research in the field of inflammatory bowel disease has helped me feel like I am contributing, even if it's just a little bit, in the understanding Crohn's and colitis that will potentially have a role in our future treatment.
Here is my lab notebook. So as scientists, we are required to keep records of everything that we do. So we have protocols, instructions, how we managed to do something, if anything went wrong, or if we just made some observations. This is a quick little tour of half of our lab. This is what we call our bacteria side, because this is where we culture our bacteria. So we have our freezers, we have a fridge, we have our chemical hood, and then on that side we store all of our chemicals. On the other side we have our big centrifuge on the bench. And as we move on, we have more of our chemicals, and then our anaerobic chamber. This is where we keep a lot of our bacteria that are anaerobic because they cannot grow in the presence of oxygen. So in order for us to get things in and out, we use this small doors, and they are connected to a couple of gas tanks. And then we have another incubator, our biosafety cabinet, and then we just have some more bench space. Here I'm showing you some of the things that I did today. So we had new bacterial strains that we needed to freeze down, and we basically freeze them down in a very small tube so that we can have several different samples of them. And usually, you have to label all of your tubes. So instead of labeling them all by hand, I like to print out on some sticker paper, and then that way that makes it a lot easier for me. So I just end up getting these stickers and then sticking them onto the tube, and so that's what I'm doing here. I then moved to the anaerobic chamber, and so these are the tubes that you saw previously. I had some bacteria that was growing for a couple of days before this, and I ended up having to mix it with a special glycerol solution so that the bacteria stay alive even when they're in the freezer. So here I am mixing them together, and then I will take small aliquots of this mixture to put into each of the tubes so that we have all the samples ready in the freezer. Lastly, before I put all of these vials in a box, I have color-coded caps that I put on these tubes so that it makes it a lot easier for me to identify them when they are still in the box, since they will be going into the -80 freezer, and it is a little bit hard to kind of rummage through these boxes whenever you're looking for a specific tube. So after I did this, I put them in a freezer box so that they will be ready to stick into the freezer. Then after that, we need to label our box so that we know what's inside of it. And in this case, this is more of a temporary label, since I will be distributing all of these samples into other boxes that we already have. In our case, we also have a separate student office, and we usually spend a lot of time here when it comes to analyzing our data or just doing some readings. This was just a brief intro of my research and some of the things that I do on a daily basis. Thank you guys for watching.